So if I could invite Dr. Brian Dickie to give the first presentation, and just a few words of introduction for Brian, who graduated with a PhD in neuropharmacology from the University of Wales College of Medicine. He then took up a research fellowship in the Department of Pharmacology, the University of Oxford. And since 1996-97, Brian has worked for the MND Association as Director of Research Development. So you give a warm welcome to Dr. Brian Dickie, please.
Thank you. As you'll guess from my accent, I'm not Welsh. In fact, I did my first degree just up the road at Glasgow University, and I had a flat with no central heating and very, very large windows just off Argyle Street. Oh, I miss that place so much. So I'm going to just talk a little bit about the causes of MND. Just to check you're all awake at the beginning, hands up who's seen the film Sliding Doors. Yep, quite a few. One of my favourite films. For those of you who haven't seen it, it stars Gwyneth Paltrow, and it's basically two films in one. And in the first version of the film, she's running for an underground train in London, and she gets there just before the doors close, gets on the train. Second version of the film, she's running for the train, the doors close just before she gets there. And that one incident sends her life off in two different directions. And it's not unusual when you're given a diagnosis like MND to think, why me? Why did I get this disease? And it's often followed by what was the thing that caused this MND. And, you know, if there was a single cause of MND, believe me, we'd have found it a long, long time ago. It's much more complex than just a single sliding doors moment. The analogy I often use is it's maybe more like a set of balancing scales, like the scales of justice, and something has to tilt the balance in favour of the disease occurring. And we know that there are many different factors that are involved here, and that's why we call it a multifactorial disease. So rather than just a big weight on the scales, single big weight, it's like lots of grains of sand on the scales. Some of those we will be born with. They're susceptibility genes. The grains of sand are sitting there in the scales, but the scales don't move. So you have to go looking at maybe environmental factors. You know, maybe you were dropped on your head as a baby, could be a grain of sand. Between the ages of 5 and 10, you lived in the countryside and Farmer Giles was chucking his DDT over the crops every summer, another grain of sand. You were very sporty and athletic at school, played a lot of rugby, did cross-country running, another grain of sand. You went to university, you did the complete opposite. You drank like a fish, you smoked like a chimney, you didn't get out of bed till 2 o'clock in the afternoon. I don't know, maybe that was just me, actually. But the point is that it could be this accumulation of different, apparently unrelated and innocuous things that eventually tilt the balance in favour of the disease occurring. And we have to add to that ageing. We have an ageing nervous system. The older you are, the more likely you are to develop MND, and that's probably linked to the fact that the body is less able to cope with whatever is loading up that side of the scales. But I'm going to focus mainly on the genetics today, but you'll notice that there is another side of the scales as well. And one of the most common questions I'm asked is, Stephen Hawking, how come he lived for over 50 years with this disease? This is my favourite photograph of Professor Hawking, you know, not a wheelchair in sight. You know, this was somebody who didn't let the disease define him. And he asked me that question himself one time, and I found myself going, imagine a set of balancing scales, whereas in my head I was just thinking, I'm talking to Stephen Hawking, I'm talking to Stephen Hawking. But, you know, maybe, as I said to him, there was something in his genetic makeup that hadn't stopped the disease, but was pushing back just a bit and slowing the progression. And so there's now increasing interest on that side of the scales as well. Because if we can identify, let's call them good genes that slow the disease down in some individuals and work out what they're doing, maybe you can develop a drug that acts as a bigger weight on that side of the scales and would be effective in all forms of the disease. So before I delve into genetics in too much detail, I'm going to give you a very simple example of how genes and environment come together to cause an effect. So hands up again, have anybody who's ever heard that the Japanese can't hold their drink? One or two, not too many. Well, it's actually not a myth. It is true. A large proportion of the population of Japan are not very good at holding their alcohol. And the reason for that is because about 50% of the population of Japan carries a mutation in a particular gene. It's called the ADH gene. And ADH stands for alcohol dehydrogenase. And as the name suggests, amongst other things it does in the body, it's a liver enzyme. It breaks down alcohol. So in these people, the genetic code, the genetic instructions to make this enzyme in the liver are slightly wrong. So the enzyme's put together wrong, and it doesn't do its job properly. And as a result, you have a drink, your blood alcohol levels rise, but they don't go down as quickly in these people. So they have another drink, they get higher, they have another drink, they get higher. I think it's no coincidence that the Japanese invented karaoke. Because, let's face it, you do have to have a certain amount of alcohol in you before you think that picking up that microphone is a good idea. But there is a very simple example. One genetic factor, one, the ADH gene, one environmental factor, alcohol. And of course, it's not just a case of, you get drunk, you stand up, you sing badly, and you fall over. The 50% of the population with this gene mutation does have long-term health problems as well, particularly heart disease and liver disease. But there we have single gene change, single environmental factor causing an effect. MND, much more complex than that. As I said, many, many grains of sand. So one question is, well, can we get an idea of what the proportion of genetic factors to environmental factors are in MND? Quite difficult to do, but one of the ways you can do that is through what are called twin studies. Now, twins are pretty rare. MND is pretty rare, so the one study that's been done does carry what we call large error, margins of error. But basically what you do is you take identical twins, and identical twins, of course, share 100% of their DNA, and you also take non-identical twins, who share 50% of their DNA. And if the first twin has a disease like MND, you ask the question, how often does the disease then appear in the second twin? And if there's a difference, if it occurs more often in the identical twins than the non-identical twins, it gives you a rough idea of what the proportion of genes to environment is. I hope that made sense. The bottom line is that this study that was done by a group in London showed that the breakdown appeared to be about 60% down to your genes, 40% down to environmental factors. Another way of looking at it, that researchers have looked at it, is how many steps would it take to develop MND? And this is an approach that's been used in cancer quite a lot, but up until a few years ago had never been used outside of cancer. But a neurologist, Amar Al-Chalabi, along with an epidemiologist from London, Neil Pearce, and some of the panel sitting beside me today were involved in this study. And they took this formula that had been used in cancer, because of course many cancers involve genetic predisposition and exposure to environmental factors. That's the formula up there in the top right. Don't ask me to explain it. I remember when Professor Al-Chalabi, we were sitting in a coffee shop, and he's scribbling this on a napkin, and he's going, it's really, really easy. And I'm just thinking, nod and smile, Brian, nod and smile. But they took this formula, and they applied it to a register of people with MND in the southeast of England. And what they found is when you took the logarithm of the age at which people developed the disease against the incidence, how many people at that age were developing the disease, what you found was everything seemed to line up in a straight line. So they said, well, okay, that's interesting. Let's go to Scotland, because there's a very nice MND register running in Scotland. And they saw exactly the same thing. And in Ireland, and in Holland, and in Italy, exactly the same line. And a straight line like that means that, and the slope of that line actually tells you how many steps are involved, how many grains of sand potentially have to load up one side of the scale. And in sporadic MND, it seems to be 6 steps. Now, just in case you think, well, if you do that for any disease, you get a straight line, you can see multiple sclerosis there by comparison. There's no line whatsoever. So we think that there are 6 factors, events, have to occur. The problem is we don't know what they are. There's been a lot of work done, a lot of epidemiology done in trying to identify environmental factors for MND. When I first joined the MND Association, we'd completed a piece of work where something like 800 people with MND had completed a 47-page questionnaire asking them about every aspect of their lifestyle, every question we could think about. And all of that information was fed into a computer at Brunel University, and we came out with one positive correlation, which was the older you are, the more likely you are to develop MND. So it's clearly not as simple as just asking a question and the answer arising. But I'm going to give you one example here, one which has been shown to be quite consistent as a risk factor for MND, and that's smoking. And I've ring-fenced where MND appears on the list of things that smoking might cause. And you can see it's about number 20 in that list. There's a vertical line there, and you see all these dots and blobs. Anything to the right of that vertical line is an increase in risk. So you can see that for MND, smoking is it's a consistent, but it's a very small increase in risk. Go right up to the top of the list, lung disease, lung cancer, and you're right off the edge of the screen there. So you have to take these things in proportion. You know, I think all this slide really tells us is, A, smoking is not a cause of MND. It might be a very, very tiny grain of sand on that side of the scales, whereas for lung disease, of course, smoking is a large weight on that side of the scales. So I'm going to spend more time talking about the genetic side because you know, this sort of work, trying to identify causes of MND, is a bit like looking for a needle in a haystack. But with the genetic side, we know the size of the haystack. It's these 3 billion base pairs, 6 billion letters of genetic code that we find in every cell in our body. So we know the size of the haystack, and there's certain things we can do, maybe to try and reduce the search. For example, can we identify where in the haystack the needles might be? And if we can find those, can we pull them out so we can examine them to see whether they're needles or not? And that's basically the approach that's being taken, thanks to some amazing advances in gene hunting technology. So you go into a genetic research lab. This is the Sanger Institute in Cambridge, and you don't see any Bunsen burners or test tubes. You see these rather large machines that are just churning out genetic code from DNA samples. And you also need some pretty large computers. A single human genome will clog up the hard drive of your laptop. So when you're looking at thousands, tens of thousands, even more, you need some pretty big toys. This is the Surf Sarah computer in Holland, which is being used, amongst other things, for, well, one of the other things is actually it's used by the European Space Agency for mapping galaxies, but it's also being used for a big international MND gene hunting project called Project Mine. And I want to tell you a little bit about Project Mine and about these two gentlemen, Bernard Muller and Robert Jan Stewart, who were both Dutch entrepreneurs who were both diagnosed with MND about the same time. And the story, as Bernard tells it to me, is that they went to their local care and research centre, which was the University of Utrecht, and they were being shown around the labs, and they were shown into this room where there was this big freezer. And the freezer was full of DNA samples. And Bernard said, so what are these for then? And he was told, oh, not for anything at the moment. These are for later. You know, it's very complex stuff and we can't afford to do it. And he said, so a lot of the answers to our disease are lying in this freezer. Yeah, and quite a few other freezers around the world. well, shouldn't we be doing something about it then? And out of that, Project Mine was born, and it now involves 19 countries around the world. I think it's a great example of crowdfunding. They've set themselves a very ambitious target of sequencing the genetic code of 22,500 people, 15,000 people with motor neuron disease, 7,500 people who don't have motor neuron disease, because of course you've got to compare the genetic makeup of somebody with the disease to somebody that doesn't have the disease. We came in very early, at a very early stage. We had a target of about almost 10% of that international target, and we're just about to finish that. And you'll see that graph on the right. I'm not going to go into that in any detail. Just see the line that's kind of rising very rapidly. That shows the number of genes that have been identified since the first key breakthrough was made back in 1993, and basically it's rising exponentially. We're finding new genes, you know, almost on a quarterly, monthly basis even. So we're really starting to get the pieces of the jigsaw that will be necessary to understand what's driving this disease in the first place. However, that's really focusing on what we call sporadic MND. You know, it pops up here, there, for reasons we still don't completely understand. But of course, as many of you know, sometimes the disease does run in families. In 5 to 10% of cases, there's a strong family history of the disease. And in that case, there must be a genetic factor that's acting more like a larger weight on the scales if it's being passed down through the family line. But you'll see there, I haven't said familial MND is a genetic disease. I've said it's a mainly genetic disease and added a question mark because it could still be very variable. Other grains of sand could be required. So some researchers just a couple of years ago thought, well, okay, let's take that mathematical model that we used for sporadic MND and let's see what happens if we apply it just to people with specific genetic mutations and a family history of the disease. And what they found, they looked at 3 particular genes, C9orf72, which accounts for about probably a third of all familial MND cases, SOD1, which accounts for about 20% of all MND cases, and one called TARDBP or TDP43 counts for maybe 3–4% of all cases. But what they found was, once again, they got straight lines occurring, and the slope of those lines tells you how many steps are involved. And you can see that there's 3 steps for C9, 4 for TDP43, and 2 for SOD1. So it's fewer than 6 steps. So the genetic factor is a kind of bigger grain of sand, a bigger weight, but it's more than 1. There are other things that still need to occur. And that explains, you know, for example, in a family with a SOD1 mutation, why one family member may develop the disease in their 30s and it progresses very quickly, but another family member may develop the disease in their 60s and it progresses more slowly, or a third family member may carry the gene and not develop the disease at all. So it's not just down to the genes alone, or at least those specific genes alone. So I've told you a lot about genetics. You know, I guess one of the questions is, well, is this all an academic exercise? You know, what does this mean in the real world, and how is this going to help us to develop effective treatments for this disease? So one of the ways is that if you've got something that is priming the disease, a fairly large weight on one side of the scales, like these hereditary genes, then we can actually take that knowledge, go into the laboratory, and try and develop laboratory models. Now, you can take the kind of deconstructionist approach, break everything down into its individual little parts, or you could look at the organism as a whole. There's no ideal laboratory model here. Each one has its pros, its cons, and each one is better for particular types of research. But we have a whole range of these. So from cells grown In addition, isolation, and you're going to hear more about that from a couple of our speakers later on, to very simple organisms like these nematode worms. Fruit flies are very popular with many researchers. You move from invertebrates to vertebrates, so zebrafish now have a rudimentary spinal cord, so you're getting closer to the human situation, to the rat and the mouse models as well. But, you know, having that starting point, a genetic factor that is really driving the disease in some cases allows you to actually develop similar models in the lab and actually find out what's going on, particularly at the very early stages of the disease. Because, you know, if we want to develop effective treatments for this disease, chances are we are going to have to try and catch it quicker in future. Another example is, of course, now, particularly if there's a family history of the disease, younger family members might think about taking a genetic test themselves. This happens in Huntington's disease and breast cancer, and particularly if they want to start a family themselves, they want the reassurance that even if they carry the gene, they won't pass it on to their children. So now you can apply for a process called pre-implantation genetic diagnosis to the Human Fertilization and Embryology Authority. And basically, this is an in vitro fertilization process. But of course, there's no fertility problems here, so it usually only needs one attempt. And you take sperm from the father, eggs from the mother, fertilize them. And when the embryos get to this kind of 8-cell little ball stage, you can pinch off a cell, it doesn't harm the embryo, and you test that cell to see if it carries the faulty gene. And of course, if it does, you set that aside. If it doesn't, then you implant that embryo into the mother. And the first time this process was used is probably about 6 years ago now, I think, in the UK, specifically for motor neuron disease. And I love this quote, which I pinched off of the website at King's College Hospital. It's from the mother of the child that was conceived. Our son was the first child in the UK conceived through PGD for motor neuron disease. He's brought a lot of joy to the whole family. It's just amazing to know that he's okay and he won't go through this horrible illness. It's a blessing to know that for our family, this illness stops with this generation. And that still sends shivers down my spine. You know, the fact that we have so few victories at the moment against this disease that I think we need to celebrate opportunities like this. And, you know, as we discover more genes, clearly that gives more opportunity, at least for a process like pre-implantation genetic diagnosis. And then I think the really exciting stuff that's really just coming over the horizon, which Professor Shaw is going to talk more about, is that if we have got bad genes that are doing bad things, can we remove that weight? Can we switch off these genes? And I'm not going to say any more about that, apart from its approach at the moment, which is called antisense therapy. And it's being trialled at the moment for the SOD1 forms of MND, and hopefully not too far into next year, we might have it trialled for the C9orf72 form of the disease as well. And, oh, there we go. And, you know, there's a lot of hope behind this technology, and that hope comes from a childhood motor neuron disease called spinal muscular atrophy, where we've seen some quite dramatic improvements in treatment recently. And I just pulled this. This came from The Guardian about a year ago, where, you know, I think for many, many years, we've been thinking this, you know, this is too tough a nut to crack. Suddenly, this breakthrough in another type of motor neuron disease does have its differences, but is really, I think, galvanizing a lot of excitement that we might actually be able to make inroads into treating other forms of MND and also, indeed, other neurodegenerative diseases, Alzheimer's disease, Parkinson's disease, etc. This is my last slide. You know, I spend a lot of time talking about advances in research, and it's complex stuff, it has to be said. The media like to distill everything down to, you know, problem, solution. I call it the rule of Star Trek. If you ever watch Star Trek, in the last 5 minutes of the program, somebody goes, I know, let's reverse the dilithium flow through the phase energizer, Captain. It's always better in a Scottish accent as well. And make it so, and in the last 5 seconds of the programme, the ship's saved from blowing up. You know, if only it were that simple. It is a far more complex problem, but I do believe that we are starting to move more in the right direction, maybe taking more steps forward than we are taking backward. So I hope I've more or less kept to time. Thank you very much.